Alright everybody, our next short video for this module is dealing with what we call the central dogma of biology. And the central dogma is kind of the central principle of how biology really works. It's the idea that all of the information stored in the DNA blueprint of organisms is transcribed into a different form and translated ultimately into proteins. And the proteins made by specific organisms give them their character. It's what we've talked about in previous modules as the difference between a genotype, in whatever's in the genes, and a phenotype, sort of that expression of those genes. And the central dogma of biology has two key components. The first is transcription, which is the focus of this video, and the second is translation, which is the focus of the video in the next part of the module. So again, DNA is the blueprint. DNA, whatever is encoded in the DNA of organisms, is what ultimately builds proteins. So we're going to focus on this part today. DNA template, transcription into mRNA. So you're taking DNA language and turning it into mRNA. And the importance here is that it's only a piece of the DNA. The entire strand of DNA, the entire genome of an organism, is not transcribed. Transcription is only a small part of the DNA, and we call this small part of the DNA a gene. So again, let's focus just on transcription for now. So transcription is the process of taking a DNA molecule, a short section of it called a gene, transcribing it into a different language, the language of RNA. Remember, DNA and RNA are related molecules. They are both nucleic acids. So you're taking one form of a nucleic acid and turning it into a more mobile form of a nucleic acid. Transcription is a polymerization reaction. And because it's a polymerization reaction, as I've said before in prior videos, it has three components: an initiation, what starts it, an elongation, what makes the molecule bigger, and a termination phase. So transcription has initiation, elongation, and termination. So let's start with initiation. Initiation is what tells certain molecules within a cell to get things going. What starts the process of transcription? Again, there are a lot of terms here, so make sure that you're taking note of particular terms and equating them with the proper activity. So here we have initiation of transcription. The initiation of transcription in any organism starts at the promoter, which is here. On the screen you can see something that looks like a guitar pick. That is where an enzyme called RNA polymerase binds. The promoter is this green section of a gene. Remember, a gene is a piece of DNA. And at the beginning of a gene is a section of DNA called the promoter. The promoter tells the RNA polymerase, which is this enzyme shaped like a guitar pick, to land on it. And once this happens, the process of elongation can occur. So initiation is when RNA polymerase lands on the promoter. Once RNA polymerase is on the promoter, it creates a small bubble. So RNA polymerase is an enzyme that creates a polymer of RNA. Remember, take the enzyme and look at the wording and figure out what it does. So RNA polymerase, an enzyme that makes RNA. Part of its job as an enzyme is to unwind DNA, but only in a very small area. So you've got unwound DNA inside of the molecule complex of the RNA polymerase. So here's a zoomed in picture of this. You have the RNA polymerase, the guitar pick, again, popping those hydrogen bonds open so that you get DNA opened up. And why do we want to open up DNA? That's so that we can make RNA from the code embedded in the DNA. So the codes here are blue for DNA and red for RNA. So we have transcription occurring in the 5' to 3' direction, just like another process we talked about earlier. So again, polymerases work in the 5' to 3' direction. And because we're only making a shorter molecule, an mRNA, single-stranded messenger RNA, we only need one RNA polymerase. So the RNA polymerase starts working. It works in a very similar way. It's using complementary base pairs. A's bind, in this case, to a new base, uracil. So A here binds to U here. C's and G's still bind together. RNA nucleotides are hanging out in the area that transcription is happening. So as the RNA polymerase moves along, a new nucleotide for RNA is brought in. It is complementary to whatever's there on the DNA. And why does that need to happen that way? That's because we want to conserve the message of DNA, only in a slightly different language, the language of messenger RNA. So the elongation step is again this movement of the RNA polymerase along the gene. So as it moves, this red messenger RNA transcript appears. And RNA polymerase works in the 5' to 3' direction. When the end of the gene is reached, there is a section of the DNA called the terminator. When the RNA polymerase reaches the terminator, it falls off of the DNA. You'll see here, you've got this dark blue section of gene, that's what's being transcribed, and it falls off, the RNA polymerase does, afterwards. And what you're left with is an RNA transcript here in red. This is another way to look at it. I'll just leave this up for a second and you can read through and figure how these things work. This is just summarizing exactly what I talked about before. RNA polymerase lands on the promoter. It unwinds a portion of the DNA. It moves along the gene, creating a messenger RNA strand. And once the terminator is reached, the RNA polymerase falls off, the DNA winds back together, forming hydrogen bonds within the double helix. Prokaryotes and eukaryotes both do transcription. In a prokaryote, transcription happens simultaneously with translation, which we're going to talk about in the next video. The difference with eukaryotes is that eukaryotes have a nucleus. So transcription within a prokaryote happens in the cytoplasm because they don't have a nucleus. In eukaryotes, transcription happens only in the nucleus. So at this diagram here, you can see that DNA is transcribed into what's called pre-messenger RNA, pre-mRNA. The RNA has to be processed, and there are several steps outlined here in this purple part over here. The pre-mRNA has two different types of nucleic acid components. It has what's called an exon and an intron. You can remember this because introns are in the way. So introns get removed from pre-mRNA. So what you get happening is first a 5' cap and a 3' tail are added. This is because once the mRNA leaves the nucleus, it's in an enzyme-rich environment, so the mRNA could be eaten up. So you put a cap and a tail on it, and the cap and the tail prevent it from being degraded on its trip to the next stage. Spliceosomes here go through and remove the introns. Remember, introns are in the way. So these exons bind together. Now you have a complete eukaryotic mRNA, which are a series of exons, which are functional components of mRNA, with a 5' cap and a 3' tail. Now what's ready to happen is mRNA in a eukaryote can leave and then be translated later. So quickly again, I just want to reiterate, in a prokaryote, transcription happens in the cytoplasm, and there's no processing involved. In a eukaryote, because it's happening in the nucleus, RNA has to be processed. It has to be capped, tailed, and have introns removed before it can go into the cytoplasm and be translated in the next step.